We're very pleased today to welcome Dr. Alessandra Petrelli. She's an MD, PhD, tenure track assistant professor in internal medicine at the University of Milan, Italy, and an internist at Pio Albergo Trivulzio, Milan, Italy. Sorry for that pronunciation. I'm sure it was not perfect, but she's going to be talking about a multi-omics integration approach relying on circulating factors. Does not discern endotypes in childhood type 1 diabetes. Really fascinating, especially on the heels of our Tuesday talk, which I just mentioned to her with Tarun Pant from Medical College, Wisconsin. So really great back-to-back talks to compare contrast. And a little bit about her. I'm just going to read a little bio on her. Her research aims to unravel type 1 diabetes endotypes, determine the role of insulin resistance in type 1 diabetes, investigate autoimmune mechanisms of insulin resistance. And she's a TrialNet investigator and member of the TrialNet Publications and Presentations Committee, or the PPC. She has numerous international collaborations, and she's funded by the European Commission, AXA Research Fund, JDRF, and the Italian Ministry of Health. Currently, Dr. Petrelli holds a tenure-track academic position at the University of Milan and practices as an internist and diabetologist at the Cardiac Thoracic Metabolic Unit of Pio Albergo Trivulzio in Milan. I have to give that a second try. Thanks so much for joining us today, and really excited to hear about this work.
Thank you, Monica. Can you hear me?
Yes, hear you fine.
Yes. Okay. So thank you so much for the invitation, and for clarity, I want to say that this project was conducted and carried out at Ospedale San Raffaele in Milan, and now I have another affiliation, so I recently moved to another institute, which is University of Milan. So today we're going to talk about endotypes, diabetes heterogeneity, which is quite of a hot topic. It's been for quite few years now. We all know that diabetes is an heterogeneous disease, already starting from, the classical subdivision into type 1 and type 2. Typically, type 1 is a disease that was historically thought to be mainly present, and the onset mainly present in youth compared to adults. However, there's a lot of evidence that actually type 1, of course, had the same incidence throughout life. Still a small fraction, but still a fraction of type 1 patients do not show persistent positivity, actually any positivity to islet autoantibodies. There are a lot of features typical of type 2 diabetes that can be found also in type 1, like excess body weight, obesity, or overweight. There's a high prevalence of overweight among type 1 patients. Insulin resistance increases the risk of developing stage 3 type 1D. And also there are genetic variants with a lot of work from Maria Redondo that are typical of type 2 that can be found also in type 1, and the natural course and the presentation of type 1 in these individuals with genetic variants are different and are more similar to the type 1 individuals, to the type 2. On the other hand, type 2 typically associated disease of the adult, of elderly individuals. Actually, there's an increase in number in children and adolescents. A fraction of type 2 diabetes do present also islet autoantibody. DKA that typically occurs in type 1. Also, there's a relatively big fraction of patients with type 2 that have an onset with DKA, and also inflammation, immune cells, the role for T cells, which are actually, a memory compartment. So they are involved in the development of islet-specific immune response. There's growing evidence, and we have a recent paper published in iScience showing that they're relevant also in type 2 context. So these are some of the indication of the heterogeneity of the diabetes as a whole. But we have been discussing and introduced the endotype concept to the scientific communities now a few years ago, with this paper published in Diabetes Care, where we discussed how important would be to address disease type 1D heterogeneity and to identify endotypes. But what are endotypes? Endotype is a subtype of diabetes that can be defined by a specific and a distinct functional and pathobiological mechanism. So a specific and well-defined subtype of the disease with a clear etiopathogenesis. So aim of the study that I'm presenting, and that it's now a preprint, so it's available and you can read and go into the topic as well, is that diabetes is type 1 diabetes is an heterogenic disease based on epidemiological data, age at onset, rate of beta cell function decline, treatment response. And we hypothesize, due to this heterogeneity, the existence of distinct molecular mechanisms operating in type 1A development. So we performed in this study what I call a bottom-up approach to unravel diabetes endotypes. A bottom-up because in this project, we start from the biology and then we build on that and we then associate mechanism or molecular pathways and information with clinical parameters, disease outcome, genetic predisposition. So this is our study design, and to do that, we utilize of course, in collaboration with bioinformatician from the Institute, the MOFA, which stands for multi-omics factor analysis, to integrate different layers of information. So we had this cohort of 146 new onset type 1D patients. This is a pediatric cohort that was prospectively collected starting from 2018. We obtained data from the immunome, the RNA, where we perform basically whole blood RNA sequencing, and metabolic hormones with a 9-per multiplex, where we tested different hormones related to insulin resistance, metabolic dysfunction. Regarding the immunome, we performed a unsupervised flow cytometry analysis on the FACS data to obtain 46 immune subsets. Then, thanks to the MOFA, we integrated these 3 layers of information. This is a factor analysis model that provides a general framework for the integration of multi-omic datasets in an unsupervised fashion. And then at the end of the process, when we identify different factors, then we associated that factors with phenotypes, with phenotypic traits. And in particular, these were the data that were available, which were age, sex, the presence of family history of type 1 diabetes, the genetic risk score 2, the BMI percentile, the presence of DKA at onset, neutrophil count in the circulation, C-peptide level, IDA-A1C, and PI C-peptide ratio. The final goal was to identify clusters of patients that would have relate to a specific mechanism or factors. So this is.
Oh, sorry, go ahead. I was just going to ask you this when you're answering.
I can't hear you.
Oh, sorry. I just was going to ask you about the age, and you've just gone to the slide with the age.
Oh, okay, yeah.
Somehow your presentation has changed from full slide. Okay, there it is. Great.
Okay. So this is our cohort. These are 146 new onset type 1D patients. The median age is 11.4. The genetic risk score 2, the score in the median is 14.4, which is in line with previous data published in this population. The BMI percentile was in the range of the normal weight. Approximately 1/3 of our population, hold on, I put again the pointer. 1/3 of our population had an onset with DKA, which is in line with our Italian statistics. The number of autoantibody, the majority had 2 or more autoantibody. We still had 15% of subjects with 1 autoantibody. We had at the beginning of the study in the total cohort, there were also 7 individuals without autoantibodies, which then were tested for genetics and resulted as monogenic diabetes that we decided to exclude from our analysis as they were not type 1. And the ADA1C below 9, which at the 12 months follow-up, which indicates the entrance in partial remission was of approximately 1/3 of our population. So this is the first thing that we did was to identify and define the number of factors that we that explain most of the variance of the integrated layers of information within that factors. So we set a parameter which was in collaboration with bioinformatician that each of the layers, immunome, transcriptome, and metabolic hormone, should explain at least 10% of the variance. And that's what we did, and we ended up with 12 factors model. But I have to say that in the paper, in the supplementary, you will see that we tested basically all the models from 5 to 15 factors, and the results are basically overlapping. So with these 12-factor models, we see that the transcriptome explains the majority of the variance. Then comes the immunome that explains approximately 20% of the variance, and in the end, the metabolic hormones. And here you see how each factor explain a certain level, a certain percentage of the variance. So you see, for instance, factor 1 is mainly explained by RNA, by the transcriptome, while factor 2 mainly by the immunome and metabolic hormones. And this is to provide a glimpse of what type of information we can obtain by each factor. So we can we can determine and visualize the analytes that have the highest weight for each factor. This is, for example, factor 1. You see in the transcriptome, you have the list of the top 20 genes that mostly explain that factor. For the immunome, these are the top 10 features of the top 10 immunological immune cell subsets that again explain that factor. And these are the 9 hormones. And adiponectin, for instance, is the metabolic hormone that mainly that has the highest weight for that factor. So these are the information that we can get by the factor. But the most important information we would have expected was to observe, based on MOFA score plot, clusterization of patients based on these factors. So you see on the X and the Y, all the factors that are close to each other, and the color shows the phenotypic traits. So for instance, this is the plot of the age. Each dot is a patient, and the blue color are individuals above the median age, and the red color below the median age. You see that we can find any type of classification, of segregation of a certain subgroup of individuals. And that goes is the same also for other phenotypic traits that we tested: the sex, presence of family history of type 1, genetic risk score, etc. The picture is small, you can see it better in the paper, but still we cannot identify any cluster of based on phenotypic traits. we performed we were quite disappointed from the results, so we performed a numerous number of amount of analysis, of different analysis to get to the same, point and goal. So here we use the heat map to make a clusterization of patients and factors. And here on the top, you see the annotation of the different phenotypic traits. But again, you don't see any clusterization or any type of association with phenotypic trait. Another analysis was based on single layers. So we decided not to integrate these 3 layers together, but we use single layers, and we perform a principal component analysis on transcriptome or immunome or metabolic hormones. This is an example of the transcriptome. Here you see plots PCA from with base colored with the different phenotypic traits, and again you see no clusterization or segregation.
Alessandra just had a question in the chat. Did you study race as part of this analysis?
No, I have to say I mean, the race is quite homogeneous in Italy, but still, this is something we should have accounted for.
Okay, great.
So we performed another analysis based on a suggestion of a reviewer, a human, which is another type of dimensional reduction algorithm. And we actually did find only in the metabolic data set a cluster based on the UMAP 1 and 2. However, again, when we looked at the different, we colored each patient based on the different phenotypic traits, we couldn't find any trait that accounted for this separation. So our conclusion in this paper is that most factors, so based on this type of analysis obtained from the integration of blood-derived omics, we were unable to identify endotypes in the specific pediatric cohort of type 1D patients. So we have discussed a lot in the paper the limitations of our study. One is the age range, because we didn't have children with age below 5. And that's because there were, technical problems for the blood withdrawal. So they were basically, we couldn't, collect blood samples, biological material by then. So this is a limitation, because we all know that the pediatric type 1D of very small children is different compared to older one in terms of severity of the disease, of pancreatic dysfunction or decline of pancreatic function, on outcomes. So of course, this is a limitation of the study, and this is clearly stated in the paper. Another limitation is that we only have data from the blood. We have this extremely interesting information, but still are from the circulation and not from the target sites of autoimmunity. And then another one is that probably other phenotypic traits could have, that were not included in the study, could have accounted for differences, or other omics. And I wanted to start from this concept to discuss a bit more this paper within the context of the literature. I mean, this project started in 2018, so there were data that were not available at the time. But for instance, if it was now, I would have included also parameters regarding the exocrine pancreas function. We and others also showed that it is impaired in relatives of patients with type 1 diabetes. But in this paper in particular, we showed that this occurs in adults. You see that there's a reduction of pancreatic amylase already before, the stage 3 type 1 diabetes, but already in individuals who are relatives without autoantibodies. This occurs in adults, but not in children. And another interesting information regards the more innate immunity, the innate inflammation where we find, this is unpublished, but platelet neutrophil aggregates that are increased in preclinical stages of type 1 diabetes, but only in pediatric individuals and not in the adult cohort. So these parameters would have been helpful to be included in this project. And also regarding other omics that may be of help, I want to mention this interesting, very recent paper from Dotta's group in collaboration with the Nodian Network, where they find that circulating mirinome stratifies individuals into 2 subgroups. These again are over 100 type 1D new onset pediatric individuals, and the mirinome analysis was performed in these individuals and clearly segregates 2 groups, cluster A and cluster B. They also do not find clear association with clinical characteristics or with phenotypic traits, except for the reduced need of insulin in follow-up visits of one of the 2 clusters that was associated also with the immunological signature of CD8 and CD4 T cell subsets. So I want to also include in the discussion this very interesting paper to me. Interesting also because they use a completely, I would say an opposite approach to get to the same goal compared to the one we use. So I like saying that we use the bottom-up approach, starting from the mechanisms, from the biology, and then associate this factor to the clinic or phenotypic trait. They started from the clinic. This is a very big cohort of type 1D pediatric individuals, over 11,000 in over 100 subjects that were enrolled, and they used the regression tree analysis that stratifies fasting C-peptide levels and identified age, H1C, A1C, and BMI as split parameters that allow the classification into 7 groups of autoantibody positive individuals and 3 groups of autoantibody negative. And then afterwards, they associated with other clinical traits, phenotypic traits, with also immunological parameters like cytokine level. And they do find some association, but still the conclusion is that there is no clear evidence of a specific pathway that is associated with a certain phenotypic trait. And then I cannot mention the recent review by Redondo and Morgan, where they state the existence of 2 type 1D endotypes, type 1D endotype 1 and type 1D endotype 2. These are age dependent. This is shareable. And here they provide evidence that in the endotype 1 is typical of individuals of very young age, below 7, associated with GR4, with IAA and IA2 as first autoantibody that presents at onset, and then high percentage of islet infiltrating cells, B cells, CD8 T cells, high beta cell stress, low percentage of insulin containing cells. On the other end, the endotype 2, which is characterized by the presence of gut and low level of infiltration in the pancreas and high percentage of insulin-containing cells. This makes sense. There's literature on that. I have to say that these are all single reports that associate age to a single parameter that may be immunology related or a clinical or phenotypic trait. What is missing to me is a comprehensive study where different phenotypic traits really converge into a single mechanism, pathogen that would explain the pathogenesis of that endotype. And I was interested in this part of the review where they discuss how the type 1D and the type 2 is still very heterogeneous. So it includes the LADA, the slow progressing, the type 1D, and also a type of diabetes that I'm very interested in and have been working over the last year, which is so-called double diabetes, where type 1 and type 2 basically characteristics coexist with autoimmunity that coexists with insulin resistance. And we have also, work on this topic. We found that insulin resistance in the big TrialNet International Cohort predicts the progression to stage 3 type 1 diabetes. There's, insulin resistance at the study entry predicts the transition and the progression to stage 3. So there may be different mechanism and reason why this occurs. It can be due to the so-called accelerating hypothesis. So in a context of islet autoimmunity, individuals with insulin resistance have an acceleration of the progression of clinical diabetes, or there may be a link between one and the other, a mechanistic link that may make an endotype. This was an hypothesis that I put forward. There's this thing that doesn't allow to see. That I put forward few years ago, where I saw diabetes as a continuum model with two extremes, where one extreme was autoimmunity based and the other one insulin resistance, and in between this double diabetes, or what I call autoimmune diabetes, where the two entities would have a mechanistic link. My hypothesis was that insulin-specific T cells would develop under a metabolic stressful condition due to overweight, to insulin resistance, to hyperinsulinemia, and the islet-specific T cells would have a role both in the targeting of T cells, but also in maintaining, sustaining chronic inflammation in insulin-sensitive tissues. I don't have mechanistic data showing that, but this is a very preliminary observation where we find that islet-specific T cells are enriched in the circulation of patients, of individuals at risk of developing type 1D without antibodies, or relatives without antibody. We see the central memory gut-specific CD8 T cells are increased in individuals, in relatives of type 1D patients with high insulin resistance. They correlate with DOMA, they are more activated, and also this is quite intriguing, insulin-specific CD8 T cells are increased in relatives without immunity and high insulin resistance. They have a naive phenotype and not an effector memory one, and they correlate again with OMA. So again, this is not a mechanistic link, but still provides data to support the idea that a link between autoimmunity and insulin resistance exists, and that it should be further investigated. So this is, an overall conclusion on the topic. My view is that there are no conclusive data yet on the existence of endotypes. There are scattered reports that associate phenotypic traits with molecular, genetic, immunological data, but still, validation is missing for some of these elements. Validation cohorts, big cohorts where different phenotypic traits really converge into a certain mechanism or molecular pathway that can somehow explain the pathogenesis. Use of integration of data integration analysis may be important in this context. I would suggest really to stress on the association of traits that would be, would predict the responsiveness to treatment, because if a certain drug that targets a certain pathway is associated with a certain trait of individual, certain characteristic, that then we could go ahead with selecting and designing trials that are specific for the type of individuals. And then, of course, the importance of the study of a target organ. I can tell that now more than before, after the evidence that we have from this data. Of course, it's a limitation that all investigators have. However, I was thinking that maybe a possibility, probably someone is already doing that, but like the endoscopic ultrasound guided fine needle biopsy in big centers with a lot of expertise may have very minimal side effects and may be more acceptable for individuals, candidates, patients, but also the ethics committee. So we really need the target organ from living donors if possible, and we need to share samples and information. That I think it's crucial. So I want to thank, of course, all the collaborators for this project and the funding agency and the generous patients and families that shared their samples and their information, and of course current and past lab members. Without them, this work couldn't be possible. So thank you so much.
Thank you so much, Alessandra. That's really fascinating. And I have to say, I've heard this on the street. It's sort of gaining more following, or at least more voice, this whole idea of insulin resistance meets autoimmunity. People are talking more about it. And so it's great to hear you actually come out and say it and walk us through the historical data. I had a couple of questions here's a few questions, then I have a few. In your opinion, what are the best ideas out there to better evaluate responsiveness to treatment? That's from Alexis Karmer. She's also an MD.
To better evaluate this responsiveness to treatment.
Right, because you tapped on the whole idea of like, okay, we have
So I think it's
endotypes.
crucial because, of course, if a drug is targeting T cells and there's a subset of individuals who respond best, of course, this should be investigated. And that individuals should be selected. I mean, the mechanism should be addressed, and that individuals should be selected for clinical trials that are dedicated to that individuals. So I'm thinking to the teplizumab and the association with exhausted T cells. I mean, this may be one of the entrance parameters to the criteria, the selection criteria to enter a new trial with that. And same would go for age, or all the traits that have been shown to be more effective in a certain trial.
Yeah, and it does really make sense, right? It's already been done in the breast cancer realm. At first, they just threw anything at breast cancer. Now it's very targeted and very bespoke. Yes. And that's where this can also go, I think, given if it's given the right attention, the amount of data it needs, and of course funding. Can I ask you, what do you think about taking Dexcom data in relatives of the T1D patients to add more data to your sort of insulin resistance AI hypothesis? How might that dovetail into what you're talking about?
Yeah, I think there are studies ongoing because, for instance, in TrialNet, there are CGM, subgroup of individuals with CGM, and there are papers investigating that. I think it's great. I mean, what we have been using as a parameters at the moment are HOMA, Matsuda. I'm working with a colleague, Galderisi, from New York, also on other type of metabolic parameters, which are the oral minimal motor derived parameters to really better study the role of insulin resistance, because the gold standard would be the glycemic clamp that is not currently performed. So the continuous glucose monitoring and this information may, of course, add to the story. Yeah.
Yeah, that's great. It just seems like it would be a natural way to gain more data. What do you think about going back into larger and older data sets to sort of be reanalyzed in context of this idea, this hypothesis,
Yeah.
and using the newer tools that you've discussed?
Yeah, so this is also ongoing because as far as I know, within TrialNet and Inodia, there are grant funded with this goal. So to go back to clinical trials already performed, but also to the TN01, for instance, so the largest screening trials to reassess with other type of parameters, metabolic parameters, the profile of these individuals. And I have a project ongoing where I'm going to try to understand the dynamic of insulin resistance and autoimmunity, autoantibodies, but also CD4 and CD8 specific T cells, islet specific T cells. This is an ongoing project together with TrialNet. So it would be interesting to see what comes first, autoimmunity or insulin resistance. And of course, then we need mechanistic studies in mice model probably to address whether there's a mechanistic link.
Right. Yeah, whether or not the alpha cells are going offline first, right? Maybe they go offline first, sort of stealthily undercover. Then you've got this huge hyperglycemia that's toxic to betas, and then the domino effect.
Yeah, we show in this paper, I didn't show the data, but that it's expected that in individuals with high insulin resistance, there is hyperinsulinemia.
Yeah.
I mean, it makes sense. And this means also higher beta cell stress, release of autoantigen, stimulation of autoimmunity. So there may be a link that should be investigated.
Yeah. Well, it'll be really interesting for, for people to listen to the two talks we had this week, yours and the one from Tarun Pant at Medical College of Wisconsin. His talk was monocytes differ in T1D families, exhibit high cytolytic activity in subset abundances. So he showed that really, there's a correlation. The monocytes of the T1D family members hypersecrete cytokines in response to LBS and ligated TLR4. So they really were acting differently at a mechanistic level. So that's kind of interesting. So he's kind of like mining a different side of the endotype. But I think it's so important that people are looking at these data, they're going after the endotypes, because as you remarked, it's really going to be important for drug development.
Exactly.
I had one last question. I loved that idea that you floated at the end, really some very specialized guided biopsy to really look at the tissue, the organ. Who do you know, or do you know of any groups who are doing this sort of ultrasound guided, very specialized biopsy?
This is a common procedure that is performed to study the pancreas. In general, typically in patients with cancer, for instance, whatever cystis or whatever is present in the pancreas. And there are a lot of centers with endoscopy units that are specialized in that. I can mention Ospedale San Raffaele, where I've been working, but I'm sure there are a lot of other centers worldwide. So it would be enough to set up a collaboration with these people and try to involve type 1D. That's always the difficult part, the regulatory aspects, and of course the willing of a relatively healthy, if it's at risk individual, a relative without antibody, or a type 1D individual, so in a stage 3 or later, even stage 4, to really donate for research, because here there's no gain at all. However, this technique, you will get cells, as far as I know, so probably not a real biopsy. However, there will be even enough to try to investigate, immune infiltration, exocrine pancreas, islet cells. You can be lucky. I mean, there may be limitation. You can be lucky that you get the islet cells, or not, because islet cells represent 1 or 2% of the total area.
Yeah, it's a problem.
I mean, there can be a lot of discussion on that, but still, we need to get samples from the target side of autoimmunity.
What about sampling? What about live sampling from the PLN? Is that even a thing?
Yeah, so we did it together with Manuela Battaglia. I mean, she did it, and I was part of the project. That was doable because that was done. Still very complicated because you need, of course, a surgeon. There are still some potential complication related to bleeding, but it can be done. She had nice data on a subset of T cells, but still, you can get few number of individuals. So to identify endotypes, I really think that we need big cohorts and data shared with several investigators.
Yeah, Jack Verosco in Texas is trying to push the needle on bioimaging in patients.
For the imaging, yeah.
So, and that would be very helpful too. Well, this was really great. Thank you so much. It was really interesting, and we'll keep an eye on what comes next. Just a really important piece of the puzzle is identifying these endotypes. And thank you for all the work you're doing in that realm.
Thank you so much, Monica, for the invitation, and see you.
Yes, see you again. Have a great
Bye.
rest of your evening. It's a great presentation. Bye-bye.
Bye-bye.